And our second speaker is Carol Upchurch, a grad student from Louisiana State University. And we're looking forward to hearing from you, Carol.
Good morning, everyone. Like she just said, my name is Carol Upchurch. I'm actually in Dr. Canavier's lab, who does a lot more computational work, but we work very closely with Dr. Gasparini that produced a lot of the actual electrophysiology data that I'm going to show you today. And like Dr. Moore, I'm going to be talking to you about place cells, but I'm talking about place cells in the CA1 region of Hippocampus, not CA3. Sorry. So like Dr. Moore, I'm talking about place cells. Place cells, I thought his cartoon was pretty great, but I'm going to go over mine again anyways. So we've got this cartoon with a rat on the right, and as the rat runs through this little maze, there's going to be a cell that specifically fires in each place in that little maze. So as it's running through that purple area, those are purple spikes from a cell that will respond to that little purple area. So that's its place cell, and the purple area is the place field. So why do place cells fire this way? Well, in the CA1 region receives synaptic input from CA3 in the entorhinal cortex. This synaptic input tends to look like a depolarizing hill, so it goes up and then down again as the animal is entering the field and then leaving. And while the hill might be symmetric, the firing is not. The place cells in the CA1 region tend to fire more on the uphill than the downhill. And that's what we're particularly interested in this collaboration I'm talking about. So this is data from Dr. Gasparini's lab. These are two separate experiments. They either injected a triangular ramp, so the same kind of idea of an uphill and then a downhill that you would have seen in vivo. They injected that ramp either in the soma or in the dendrites. And this is the data from that. And there's a couple of things I want to draw your attention to. So first off, I want you to look at the frequency graphs. This is the instantaneous frequency. So we took the inner spike interval here, and whatever current is right in the middle of that, it's current, then that frequency. And what I want you to notice is that the filled circles are on the way up, and the open circles are on the way down. And at each injected current, you're getting higher frequency on the up ramp versus the down ramp. This is the adaptation that I was talking about. And so that's the first thing I want you to notice. The second thing that I want you to notice is that this adaptation is more accentuated further out in the dendrites than it is in the soma. And the third thing that I want you to notice is that there's a decrease in spike height as this depolarizing wave goes on. And that's the phenomenon we want to capture in our model in Dr. Canavier's lab. So I did not start from scratch with this model. Dr. Canavier had previously published a model of a CA1 pyramidal hippocampal neuron. It's a multi-compartment morphologically realistic neuron. This is the morphology right here. It's implemented in Neuron. You can download it off of model.ub. The model accession numbers right here. It's a Hodgkin-Huxley type model. So we're looking at the equivalent circuit right here. Each of these resistors is your ion channel, and they're all in parallel with the cell membrane capacitance. We made a couple of changes to this model, and that's how we ended up modeling the adaptation. We replaced this sodium channel in the soma and the apical dendrites. This used to be a Hodgkin-Huxley type sodium model with the 3 activation gates and the inactivation gate. We replaced that with Markov model. We also added a second potassium delay rectifier, which I'm going to get to later. This is an updated schematic of the model. So again, we added a second delayed rectifier. This is because I wanted to make a point that experimentally, these currents are not uniformly spread across the entire neuron. So we didn't model it that way because that wouldn't be realistic. certain channels like IH and IA, the conductance is greater the further you get away from the soma. And some of the properties of these channels also change the further you get from the soma, and I'm going to get to that. So I'm a desserts first kind of person. I wanted to show you that we were successful. We do have the adaptation that we were trying to model. It is accentuated in the dendrites. That's kind of visible in the short ramps. This is 1 second up and 1 second down. It's more visible in the longer ramps. This is 5 seconds up and 5 seconds down. But I'm going to show you how we got to this. The first thing I want to talk to you about is the Markov model. A lot of you are probably more familiar with the Hodgkin-Huxley model of a sodium channel, the one that had 3 activation gates and an inactivation gate. That's technically a Markov model. A Markov model just means that whatever state you're going to next is dependent on your current state and not any state you were in previously. The Hodgkin-Huxley 3 activation gates and an inactivation gate is technically a Markov model. The problem we were having is that all of those gates are independent of each other. So when we were trying to implement slow inactivation, we didn't want the channel to go from open or closed straight into a deeper inactivated state. We wanted it to go from inactivated to a deeper inactivated state. So we moved to this Markov model. This was, sorry, having trouble with the slides here. 1 second. This is these 2 closed states. If you were trying to think of this as that Hodgkin-Huxley model, you'd probably think of these as the inactivated gates being open, but the one of the 3 activation gates being closed. This open state is when your inactivated gate is open and all of your activation gates are also open. This is one of those inactivation gates, and this is that second deeper inactivated state that we were trying to implement and to get that slow inactivation. There are some people who have tried to do that when you, with the Hodgkin-Huxley model, they just added, instead of M cubed H, they have M cubed HS, with S being the slower inactivation gate. The only reason we didn't like that is because then you could go straight from one of these closed states into that inactivated state, or straight from the open state to that inactivated state. And that gave us some really funky waveforms. So we decided to transition to this model. So why were we so interested in slow inactivation? Well, this is some data from the Spruceton lab. This is a series of cell-attached patch clamp experiments. These are a couple of sample traces from each. They're from different distances from the soma. And what they did is they held it with a voltage clamp at resting potential, but they delivered a 20 Hertz pulse stream. And then they were looking at the amplitude of this first spike and the amplitude of the last spike so that they could calculate a sort of stable percent inactivation that would happen the further they got into this pulse stream. And what I want you to notice is that that percent inactivation increases the further you get from the soma. And the reason we thought that this might be a mechanism for adaptation is, 1, it kind of explains that decrease in spike height that we saw in experiments. And 2, because it increases the further away you get from the soma, it would make sense that if it was contributing to adaptation, that adaptation would increase further out into the dendrites. So I wanted to model this slow inactivation. So I took the Markov model and I put it in a little patch in Neuron, the software that I was using. And I just increased this transition. I just multiplied it by a scale factor and I would simulate a pulse train and calculate a percent inactivation. If you do that, what you end up is the black line over here. I'm trying to get to the black line that is supposed to be that exponential from the last slide over here. So I'm trying to get from there to here. So I just fit that first black line with that purple line. It's a 4-piece linear equation. From there, now I know what kind of percent inactivation I want in each compartment from the distance from the soma. Now I just had to say, okay, so for 50% inactivation, I need to use a little bit more than 2. And that's what this graphic right here is. It's just showing you the distance from the soma, from each distance from the soma, what scale factor I'm trying to use. And the results of my model are this red line. I know there's a little inconsistency there, but it's well within the noise. So it was kind of good enough for my purposes. except for one problem, and that is that I was approaching that steady state percent inactivation a little too quickly. None of these traces look like that trace. That's a pretty simple problem. What happened is that both of these transitions were too fast compared to all of the other transitions in the model. They were too big. So I just scaled them both down by 0.2. As long as my I1 to I2 transition is bigger than my I2 to I1 transition, I'm going to have, as long as that ratio is bigger, I still get the increased inactivation. But if I make both of those transitions smaller, I slow down my time course. And once I did that, it was pretty simple to, refit the equation. And I had a model where I could change the slow inactivation based off of how far from the soma I was. And I took that model and I put it in Dr. Canavier's previously published model. And I got results that looked like this. I did not capture adaptation on my first try. I was really confused by this because we were really thinking, slow inactivation would be the mechanism. So I didn't understand why I got this weird burst. If it wasn't the mechanism, I still wouldn't have expected this very strange result. But when I showed it to Dr. Gasparini, she was thrilled. And why was Dr. Gasparini thrilled? Dr. Gasparini was never totally satisfied with our explanation of potassium delayed rectifiers in our model, in the previously published model. And that's because our previous published model only has one potassium delayed rectifier. In CA1 hippocampal pyramidal cells, there's actually two channels that act as delayed rectifiers. There's Kv1 and Kv2. And this is what happens when you block Kv2 experimentally. This is the same kind of experiment she gave at a ramp. So a ramp up, a ramp down, a triangular current ramp to look like that depolarizing hill that place cells receive. And the red trace is what happens when you block Kv2. The black trace is control. So I hadn't replicated adaptation. I replicated this weird bursting that happens when you block Kv2. So we just went back to model DB and we found a model of Kv2. The model accession number is right here. If you go and put that in model DB, it's going to tell you it's from the Allen Institute's data set of cell types, and it'll say it's a visual primary cortex. But if you look at the mod file itself, it was actually fit from data from this lab, and it's CA1 hippocampal pyramidal cell neurons. So we took this model of Kv2, we put it in our model, we adjusted the conductances accordingly, and this is just circling back to the results that I showed you a little bit earlier. So how exactly is that working? Kv2, when I graph the currents by themselves, tends to be a little bit more active in that hyperpolarization that happens. So this is a zoom in of those waveforms. And you can see it as it hyperpolarizes, that's where Kv2 really kind of shines. And I think you need that after hyperpolarization when we're talking about the Markov model to push the model out of this inactivated state and back to the available pool. So to really show that, I want you to look at the C1 state. So they're not equivalent. It's not equivalent to the Hodgkin-Huxley model, but if I was going to make analogies, this C1 and C2 state, the inactivation gate is open, but one of the activation gates is closed. So it's available for spiking, but it's not currently open. And I think you need that after hyperpolarization that was caused by Kv2 to push it out of the inactivated state and back into the available pool. And that's why I graphed C1 here. So this is just a victory lap. We're going to look at the experimental data versus the model, and these are the short ramps. Again, it's a triangular current ramp in the experiment, and it's a triangular current ramp that I simulated a neuron. And we did successfully replicate adaptation. There's definitely more firing on the up ramp versus the down ramp. It is a little bit more emphasized in the dendrites. That's a little bit more visible to see in the long ramps. So this is 5 seconds up and 5 seconds down. This is the experimental data. Over here we have my model. And the other thing that I want you to see is that we have that change in spike height. And that's part of why we really thought that slow inactivation could be the mechanism for this adaptive firing. So I'm going to wrap up now. We created a Markov model of Nav1.6 that could implement slow inactivation. and that could implement slow inactivation in a spatially distanced way from the soma. And when we put that model into our previously published model of a CA1 hippocampal neuron, we got bursting, which is exactly what happens when you block Kv2. So I didn't replicate adaptation. I replicated the bursting that happens when you block Kv2, which showed us what was the problem with our model. So then we put in Kv2, and that restored spiking. And with both Kv2 and the slow inactivation of Nav1.6, we were able to capture the adaptation and the accentuation of that adaptation in the dendrites and the decrease in spike height that you see in the dendrites. And I'm ready to take some questions.
Thank you very much, Carol. This is very exciting, and also detective work that you did. My congratulations. Do you have any questions, maybe from the panelists? In this case, I will just I will have quick questions. So it's surprising that in the dendritic response, you can also predict size by putting Kv2 in the soma, or I misunderstood we also have Kv2 in the dendrite. You don't really.
Kv2 is also in the dendrites, yes.
Okay, and how did you get the distribution of the Kv2 in the dendrite?
There's some immunohistochemistry results from the Nusser lab, and it shows Kv2 in the dendrites. It's more in the proximal dendrites and less in the distal ones. So I put it in the first 150 micrometers of the model, and then I multiplied it by a scale factor 0.1 because you can still see it out in the distal dendrites, but not half as much as in the proximal dendrites. So it's not uniformly distributed.
Yeah, but still gives very promising results. It's nice. Thank you very much. And unfortunately, we have to move forward. These sessions have to be